Let us come to the topic. Today's topic is sterilization. Why the sterilization is needed? It is the procedure. Sterilization is the procedure which is mostly used in the microbiology department, in the microbiology lab. So our focus is to describe the sterilization. Why it is needed is why sterilization is needed because of three reasons. These are the three reasons: the microorganisms or the bacteria. These cause the three the contamination, infection, and decay. So, in order to inhibit or in order to avert the contamination, infection, or the decay or spoilage of the foods, it is necessary to remove them from the materials and areas. So it becomes necessary to remove these bacteria or the microorganisms from the materials and the areas. In the olden days, the procedures for the to avert these bacteria or the infections or the contamination or decay, these procedures were used in the olden days. The salting, smoking, pickling, and exposure to the sunlight. These were the procedures were used in the olden days. Salting. The salting is the usage of the sodium chloride, that is known as the common salt. This is used as a procedure in order to preserve the food, to avert the contamination. And the pickling is also one of the procedure that is used for the preserving or the extending the shelf life of the food. So these were the older procedures, the older methods used as to avert the contamination, infection, or spillage of foods. Now the actual the topic, that is the sterilization. It is defined as the process of freeing an article from microorganisms, including their spores. It is defined as a process, the process of freeing an article from microorganisms, including their spores. Articles are freed of all microorganisms, both the vegetative bacteria and the both the pathogenic and non-pathogenic bacteria. These are killed by the sterilization. So sterilization is a process to get rid of the all microorganisms. And another term is the disinfection. This is defined as a process of reducing the number of pathogenic vegetative microorganisms to the point where they no longer cause disease. So there is somewhat difference in between these two terms, the sterilization and disinfection. Sterilization is freeing of an article from all bacteria, all the microorganisms. Either they are the pathogenic or they are non-pathogenic. In case of the disinfection, we get rid of the only the pathogenic vegetative microorganisms. So disinfection is defined as a process of destruction of pathogenic vegetative organisms capable of causing infection. The agent usually is chemical called disinfectant. So it is important to understand these two terms. Now, how the bacteria they are controlled, how the microorganisms they are controlled. There are the three methods by which we can control or we can inhibit their growth. These are the number one physical agents, chemical agents, and the mechanical removal. So these are the three methods. The physical methods, chemical methods, and the physiochemical methods, they are again subdivided into different methods. The most important of them is the physical methods. The physical methods are the sunlight, heat, the ultrasonic vibrations, and the radiation. Again, the radiation may be ionizing radiation or non-ionizing radiation. Now, among the physical methods, which we will discuss the heat. Heat is a very important method of sterilization. So heat, the methods of heat, the heat may be in the form of dry heat, it may be in the form of moist heat. A moist, in case of the dry heat, the examples, the methods which in which the dry heat is used are red heat, flaming, incineration, hot air oven, and infrared. So these are the methods, dry heat methods, and on the on this side is the moist heat methods. Again, moist heat methods, they are subdivided into three parts. In the three methods, those which in which the heat is applied and the moist heat is under the 100 centigrade. And second is the temperature is 100 centigrade. And third, the temperature above the 100 centigrade. So the moist heat again divided into these subdivisions. Now coming to the Individual methods. Individual methods of the dry heat or the moist heat. Physical methods. The sunlight that contains the ultraviolet rays, and these ultraviolet rays are bactericidal or the bacteriostatic. So it is one of the method. Physical method. Drying is another method. We air dry the things, such as clothes are kept exposed to the sunlight, so they are dried. The filtration, the filters are used. The water that is filtered we use in daily life. So the filtration is one of the method of escaping the microorganisms. To get rid of the microorganisms, filters are used, and thus the water is capable to be drink. The radiation is also one of the method. The radiations, they they degrade the proteins, and they degrade the nucleic acids. The sonic vibration is one of the methods of physical, and the heat. Among these physical methods, the most the method of choice is the heat. The heat is the method of choice of sterilization. So the you have to remember the heat is the most important method of sterilization, and the heat may be in the form of dry or the moist heat. These images shows the method, dry heat method. This is the red heat. This is a Bunsen burner, and you see the red heat, while this is the flaming. So little bit difference. This is red heat, and this is the flaming. Now, what is the one of the method of dry heat is the flaming. What is flaming? The materials are passed through the flame of a Bunsen burner without allowing them to become red hot. This is known as flaming. Red hot, the red heat is another method, and this is a flaming. Without becoming, without allowing them to become red hot, this is known as the flaming. And usually the articles or the objects which are sliced by the flaming, by this method, by the flaming, or the glass slides, scalpels, and the culture bottle, the mouth of culture bottles, the culture tubes or the culture bottles, they are they are they undergo the process of sterilization, that is flaming. So the next one, how the sterilization or the how the heat causes the sterilization. The heat method, the mechanism of the heat is that it causes the the reaction by three ways. One is the protein denaturation heat. What does the heat cause? The heat causes the protein denaturation of the bacterial or the viruses. And second mode of action is the membrane damage. They damage the membranes of the bacteria. And the third way is the enzymic cleavage. So heat causes damage by three ways: protein denaturation, membrane damage, and the enzymic cleavage. The, these are the modes of action of heat. The methods of sterilization by the dry heat or red heat, number one, and second is the flaming, and third is the incineration, and the fourth one is the hot air oven. In case of the red heat, in case of the red heat, the metallic objects such as the wire loops, the points of the forceps and the needles, they are sterilized by a red heat procedure. And in case of the flaming, there is a direct exposure for few seconds, and the objects that are sterilized are the scalpels, the neck of the flask, and mouth of the culture bottles, glass slides. And the third one is the incineration. And what is the incineration? That is a disposal of the contaminated materials of the labs and surgical wasted material, that is incinerated. जलाना, incinerate means the जलाना, किसी चीज को जलाना, जो wasted material है उसको हम जला देते हैं. And the fourth one is the hot air oven. Now the hot air oven. What is the hot air oven? This is one of the instrument, and they have the specifications, and these specifications are these are the specifications. The if the temperature is 160 centigrade, we allow for the time of 2 hours. So if the temperature is 160 centigrade, time is given is 2 hours. And if we increase the temperature, time is reduced. If it is a 170 centigrade, the time is 1 hour. And if is again the temperature is raised, that is 180 centigrade, the 30 minutes are allowed. So hot air oven is an method of sterilization, and the specifications are these. The time is here, on this side is the temperature, on this side is the time. So you have to memorize the temperature and the time. What are the uses of the hot air oven? Look at this. First of all, look at this. This is a hot air oven. The objects are kept inside these shelves, and this is closed, and time is fixed. and the temperature is also fixed. So the according to the table, so the time is also fixed, and we close this air oven, and the objects are kept, thus they are freed from the bacteria or microorganisms. So this is hot air oven. Now what are the uses? Uses: the glass syringes, petri dishes, test tubes, flask, scalpels, scissors, pipettes, all these are sterilized by the hot air oven. So this is one of the method of sterilization. The temperature is above the 100 centigrade. Look at the temperatures, the specification. So this was the dry method. This was the dry method. The hot air oven is a dry method, and the other dry methods were these: the red heat, flaming, incineration. And so these are the four methods of the dry heat by which the sterilization process happens. Now the next method of sterilization is the use of the moist heat. We I elaborated the dry heat methods. Now the moist heat method of sterilization. The one of the method is moist heat method is the heating under pressure of water. This is the moist heat. Moist means the water. Water is present, the liquid water is present. So the heat is type of the heat is moist heat. While in case of the dry heat, there was no liquid. That was it dry. Here the heat is in the form of the moist. So by this method, the microorganisms are killed by coagulating their proteins. The coagulation of proteins occurs. In case of the moist heat, the coagulation of protein occurs. And moist heat method of sterilization is subdivided in this way: temperature below 100 centigrade, temperature at 100 centigrade, and the temperature above the 100 centigrade. The examples of this one, the temperature below 100 centigrade, the methods are pasteurization, incipization, and hot water bath. So you have to call upon your memory, you have to memorize. These are the three methods under 100 centigrade: pasteurization, incipization, hot water bath. And the, the temperature at 100 centigrade, the methods are tendrilization, boiling. And the temperature, when the temperature is above 100 centigrade, the method is called the autoclaving, in which the autoclave instrument is used. So this process is called the autoclaving, or the autoclave. Now to discuss the individual methods of moist heat. The pasteurization, this is the term more or less is referred to the milk, the procedure that is employed in order to decontaminate the milk is called the pasteurization. The these some of the bacteria they are present in the milk. They are decontaminated. They are we get rid of them by pasteurization. So the pasteurization of milk, there are the two methods. It is the pasteurization, but the ways are two. One is the Holder method, second is the flash method. In case of the Holder method, temperature is 62 centigrade and it is it is applied for 30 minutes. So this method of pasteurization method is called the Holder method. The temperature is 62 centigrade and this is given for the 30 minutes. Other method is the flash method, in which the temperature is the 72 centigrade and this is applied for 15 to 20 seconds. So you memorize these two methods of pasteurization. The heating is always, in this case, in case of the flash method, the 72 centigrade for the 15 to 20 seconds, but the heating is always followed by sudden cooling of milk. This is the difference in between these two methods. In case of the flash method, heating is always followed by the sudden cooling of milk and temperature is this one, the 72, and time is 15 to 20 seconds, while here the time was 30 minutes. And we get rid of the bacteria which are killed by the pasteurization are Salmonella, Brucella, and the Staphylococcus aureus. They are killed by the pasteurization. So it is better to drink the milk after the pasteurization because the some of the bacteria they are killed by the pasteurization. The Salmonella, Brucella, and the Staphylococcus aureus they are killed. The hot water bath. This is one of the method of sterilization that is used for the that is used in the vaccine preparation, and the temperature is given that is less than the 100 centigrade. So the temperature is at 100 centigrade for 1 hour is used, and this is used in one of the it is one of the step in the vaccine preparation. So this is the water bath as shown in the image. The incipization. Incipization is one of the method. Temperature is below the 100 centigrade and that is 80 to 85 centigrade for half an hour for the 3 successive days. So this is one of the method of sterilization below the 100 centigrade, and the media which contain the proteins such as the serum or the egg, and the media, the culture media, they are sterilized by the incipization or the Lowenstein-Janssen and the Loeffler serum. These are the two culture media which are undergo this incipization method. This is the incipitator. This image shows the incipitator. The boiling. Boiling is one of the method that is the temperature is 100 centigrade. This method is implied the temperature that is used is the 100 centigrade. So in case of the boiling, the vegetative forms of the bacterial pathogens and the time is that is allowed is the 5, 10, or 20 minutes. It may be any form. The 5 minutes are given, or 10 minutes are given, or 20 minutes are given for the boiling procedure. And what happens? The some of the viruses they are killed. The hepatitis viruses they still they can survive up to the 30 minutes. So the viruses are not killed by the boiling, and the spores they are also not killed by the boiling. So this is not a most efficient method. Boiling is not a most efficient method to kill the viruses or the spores. We need the temperature above the 100 centigrade for killing the viruses and the spores. So this is not mm, the most favorite method of sterilization. Steam at atmospheric pressure. In this case, the temperature, the atmospheric pressure is applied, and the time is 90 minutes is used. For this method of sterilization, the steam at the atmospheric pressure, the exposure time is the 100 minutes. So this was also the one of the method. This is the steam sterilizer. Another method that is called the Tendrilization, and that is the temperature is the 100 centigrade, and the time is the 30 minutes for the 3 successive days, and and this is called the Tendrilization. Media containing sugar and gelatin they are exposed at this temperature 100 centigrade for 30 minutes for the 3 successive days used in a free-flowing steam, and this is also known as the intermittent sterilization. But the more familiar term is the tendrilization. Temperature is 100 centigrade. The most efficient method of sterilization is the steam under pressure. Now we use the temperature is above 100, up more than the 100 centigrade, but concomitantly the pressure is also applied. So along with the temperature above the 100 centigrade, the, the pressure is applied. So the heat is transferred more efficiently to the articles that are sterilized, and the bacteria they are killed more efficiently. So this is the most choice method. The most important method is the autoclaves. The best method of visualization is the autoclave because the steam under pressure, the pressure is given and temperature is also above the 100 centigrade. So this is the most reliable and effective method. So you remember the most reliable and effective method is the autoclave. The options will be there. Options will be and other options will be there in the BCQ. But the autoclave is the most efficient and reliable method. The autoclave may be in the form of the vertical or the horizontal cylinder. The cylinder may be vertical or horizontal, and this the pressure gauge is also present. So this is the image of the autoclave. This is the autoclave, and this is the pressure gauge, and this is the lid, and the heat is given by the electrical method. This is a horizontal, this is a vertical type, and these are the autoclave. Now the chemical agents. Chemical agents, these are different chemical agents which are used, and the methods and the mode of action, how these chemical agents act. These are the three: the cell membrane disruption, the modification of proteins, and the nucleic acid modification. So chemical agents act by these three ways, and these chemical agents may there are the different chemical agents. They are used as a disinfectants, or the sterilization agents, or the disinfectants. These are the alcohols. Alcohol is not used as an antiseptic in the pure form. It is given in the 70% form, and the water is also admixed. So alcohol is used in the mixed form. It is more effective, and the clinical thermometers and in the procedure of venipuncture, these alcohols are used. These these are the alcohol pad which is used for the prior to injection, and and the antiseptic. The phenols are also used as an antiseptic, the disinfectants, and the most common example is the pyodine. You will be familiar with the pyodine, which is used as a topical agent, antiseptic, and this is also used as a gargle when you have got some pain in the throat or the tonsillitis. We use the pyodine gargles. So this is one of the phenol, that is chlorhexidine, and these are the examples of chlorhexidine preparation available in the markets at the medical stores.